Oh, sorry. Okay, thank you, Sophia, for giving me the feedback. Yeah, I was muted. Okay, so good afternoon, everyone. I'm so excited to share this lesson. I've already unmuted. Can you hear me now, Sophia, Yitang? Can you hear me now? Okay, I'm excited to share this lesson with you today because this is one of my most favorite disciplines of biology, which is it has to do with taxonomy, which is classification of organism. Okay, can you hear me now, Sophia? Can I have feedback from you? Okay, Rachel. Hi, Rachel. Okay, I see a yes there, so I will proceed. All right. So this is one of my favorite disciplines of biology, which is taxonomy. Taxonomy deals with actually classification of organisms. Okay, so that means we need to put them into different groups so that we are able to study them in a systematic manner, systematic way. Okay, so without further ado, let me share my screen. Okay. All right, so this is today's lesson, which is biodiversity. Okay, let me look at this. All right. Now, what is the meaning of biodiversity? Okay, now if you know the meaning of bio, right? Bio means life or living organisms or living things, right? And diversity means differences. So when you put these two words together, it means the differences in all the living things that you see. So there, of course, there are thousands and thousands of species or different types of organisms, animals and plants, and also bacteria in this world. So we have a rich biodiversity on our planet here on the earth. So the whole thing is like an ecosystem, a very large ecosystem. Okay, so we have biodiversity, which is actually meaning it refers to the variety of living organisms. So we have three major groups here. We have animals and plants and also microorganisms. Okay, so taxonomy is the field or the branch of biology which deals with classifying organisms in specific groups, okay, and also giving a certain name. So this name is very specific. So it actually only specific for that certain type, or we call it a species. So we're going to learn in this chapter, all right, we're going to learn how to, what is the system of classification for all the thousands of organisms that we see on this earth here, and what is the system like, how to name it, and also the form, how do we name it? I mean, the system of naming it, all right? We will see examples and so on. So this, what they call the field of science or the field of biology, which deals with naming and classifying, is called taxonomy. Okay, so here is your the reference here from your book, all right? So here I've already gone through the meaning of biodiversity and also taxonomy. It is a field in biology which involves classifying, that means putting them into groups based on the characteristics, identification, that means identifying the features on it, and also naming it. Okay, so this is what taxonomy does. Okay, and why do we need to do this? Why do we need to classify them into groups? Okay, so it's a very simple concept. Now, why do we need to? Because you see, on this world here, on this planet so far, we have around about, scientists have estimated that there are probably about 8.7 million species, I'm not talking about 8.7 million individuals. Species means here the type. So alone it talks about type. There are 8.7 million different types of organisms, including animals, plants, and also microorganisms, all right, and all the small ones that you need to see under the microscope. So we're not talking about individuals. Individual means you have one type, but there could be many, many copies or many, many individuals under the same type. So 8.7 million here means 8.7 million different types of animals or plants or bacteria. Okay, so that means it's a lot. In fact, the population of animals, okay, and plants altogether, it far outnumbers the population of human, okay, on this earth. Okay, so now why do we need to classify? Why do we need to do taxonomy? All right, why do we need to carry out this classification? Okay, first of all, the name of taxonomy comes from two Greek words. One is called taxis, the other is nomos. Taxis means arrangement or hierarchy. Hierarchy means the order, whereas nomos means the method. Okay, so this notes is shared by to me by a teacher in Para who's so gracious to share his notes to all the other teachers. Okay, so I'm making use of his notes here. So it's very useful and it's quite informative, very informative. Okay, so taxonomy is a branch of biology that involves systematic classification, identification, and also naming of the organisms. So how do we name the organisms? How do we classify it? It's usually based on the properties, and usually we look at the common properties. We call it similarities. So if this organism has, normally it's we call it outward appearance. We don't look at the internal. Usually it's the outward physical appearance. So if it has got feathers, let's say organism A has got feathers, organism B also have feathers, so probably they will be put in the same group. Okay, and groups will have different names. Later on you'll see the different groupings. Okay, so like let's say another organism C, let's say it has short fur, and you look at another organism D, it has a short fur. Probably they also belong to the same group, and they do not belong into the group which has fur. It has feathers. Okay, so we look at the outward appearance. We call it physical properties. Usually it's external physical properties, and then we classify them. Okay, so the aim of taxonomy, right? What is the purpose of taxonomy? Is so that you can manage materials, so that you know where to find it. If your things are all scattered, okay, if you do not have a proper way of putting into groups, for example, you come to a house and your house is all disorderly, all your possessions are scattered everywhere in the house. When you want to find it, you'll find it's very difficult to find, isn't it? So what we normally do is you put the things that belong to the same category probably in one box. Okay, stationery put into one box, all right? And then your personal toiletries you put in another box. So it's easy for you to retrieve the information or to get the information whenever you want it. So that's called managed materials. Okay, so it's more systematic. You can study it systematically. You can also find the information systematically. Okay, another thing is all this information is collected through systematic approach. That means it has to follow certain rules. So if your organism has these properties, you must put into this group. It is not like you put wherever you want. It's not to your whim and fancy. So you have to follow certain rules when you want to classify the organism. To facilitate means to help. Okay, to help referencing. Referencing means you want to refer back, okay, among the scientific communities. That means all the scientists all over the world. Okay, what is the importance of classification? So that the organisms can be identified easily. You may be able to find out, okay, if it has got fur, probably is a mammal. So if it's got feathers, probably it belongs to the bird class. Okay, so we can see based on that, if it's in that group, then we know that it belongs to that specific group there. Okay, so facilitate is to help discussions at international level, so that when scientists they talk about a certain animal, they are all referring to the same one. Because when you talk about animals, let's say tiger, in fact there are 9 different types of species of tiger. If you say tiger, we do not know which tiger you're talking about. It could be the Siberian tiger, it could be the Bengal tiger, it could be the Malayan tiger, it could also be the albino tiger. So there are different, we call it species, and also the subspecies of the tiger. So when you want to be, you have to be very specific. So when the scientist A talk about something with scientist B, they all are talking on the same language and they're referring to the same thing. Okay, so the naming is very important. They have, let's say A and B, they may look the same, but they could have slight differences which require them to have a different name. Okay, so that's why there are so many species, because of difference of features. A little bit different, then you cannot put it as the same species. You must put it maybe in a different species, consider as a different species. Okay, so next one, to avoid confusion, like I mentioned, when you want to talk about it, you must make sure that you're talking about the same thing. You avoid confusion. You cannot have 2 organisms which use or share the same genus and species name. Okay, otherwise you'll be confusing. Then to ensure that no 2 organisms have the same name, all right, that's what it actually means. So first of all, now let's look at the how to classify. Okay, so we're looking at the top down. That means from the top first, the whole picture first. Okay, then only we go to all the little details. So first of all, if you look at your textbook, right, you have we're going to look through at all this taxonomic hierarchy system. Hierarchy system is all this order lah. So we're coming from the top. We're going to start with the domain. The domain is the highest grouping, the top grouping. One domain has got many, many species, thousands and thousands of species. That is the biggest group. We call it domain, okay? Then we also look at dichotomous keys. That will be the next lesson. And also how to name each organism. For example, humans, we are known as Homo sapiens, okay? So this is a genus name and followed by a species name. So Homo sapiens, and everyone must refer to humans with the same name. You cannot use a different name because there's only one species of Homo sapiens. Okay, there's no other species. Or even though different races, okay, we may be different races. We have Indian, Chinese, Korean, Japanese, or Mongolian, or even American, right? Or Caucasian. In fact, all over the world, humans actually only have one species, which is Homo sapiens, the modern man. Okay, we're not talking about the Neanderthals and all that. That is primitive. So now we're talking about the modern man. The species is called Homo sapiens. Okay, so we have seen why it's important to classify organisms. Okay, now we want to see all this classification. Now, there are 6 kingdoms. Like I mentioned, the highest order, right, is called a domain. There are actually 6 kingdoms, right, inside that. So here you have 6. Now, I want you to see the whole picture. So I want you to look at this. Easier for you to understand. All right, I'm going to look at all this. Yeah, here. Okay, if you have any questions, you can feel free to put in your comments. Okay, so far nothing. Right, now organisms means living things. Okay, organism purely means living things. So basically you have 2 groups here. You have what we call a prokaryote, a group that is called prokaryote, and a group that is called eukaryote. Okay, what is the difference? Now, pro means primitive, or the pro actually means before. Yeah, that means it's the ancient type lah. Okay, pro is primitive. Karyote actually refers to the nucleus. So for prokaryotes, what is the characteristic that you need to remember here is prokaryotes do not have membrane-bound organelles. That means the organelles, if they have organelles there, inside there, for example, the nucleus, you cannot draw a membrane around the nucleus. So it's just like when you want to draw the cell. These are basically bacteria. Okay, if I want to draw a prokaryote, all right, so look at this, I'll just try to draw one. Okay, this is a prokaryote bacteria. Okay, I'm drawing a rod-shaped bacteria. So if I want to draw the nuclear material inside the bacteria, I cannot draw a membrane around it. Okay, because there isn't any membrane around your nucleus. So you have to remove that line there, and you just draw your dot here. This refers to the nuclear material. You understand what I mean? Okay, so when you're talking about a prokaryote, there is no nuclear material, and the DNA is circular in form. So it's like, it's not long strands here. It is circular. It's like, you would call it a plasmid, S-P-L-A-S-M-I-D. So it's circular DNA. That is prokaryote. Okay, now let's look at eukaryote. Eukaryote, on the other hand, they have the total opposite lah. Whatever the prokaryote has, the eukaryote will be different. Eu actually means, in Greek word, it means real. Okay, real. It has real nucleus. So we call it eukaryote, means it's like something, what it means, real nucleus. So it has got membrane-bound organelles. All the organelles are inside there, there is membrane-bound. So we look at, let's say we look at the nucleus, you can draw the nucleus with a membrane, and then you just dot over here. You understand? Okay, so this is a eukaryote. And they have linear DNA. So the DNA is like what you have learned earlier. Remember your DNA material like that, right? You have double helix like that. Okay, so it is linear. It is not circular. Circular would be something like this. So you have a circular DNA. Okay, wait now. It could be double-stranded. Okay, so I will draw this double-stranded, right? But it is like circular, so it will be like that. Okay, so sorry, I'm not very clear. So it's like double-stranded, but it makes a complete circle. So this is a circular DNA. All right, okay. So under prokaryote, okay, like I mentioned, there are six kingdoms here. Okay, there are 6 kingdoms here. So we have got under prokaryote is divided into 2 kingdoms. So we have something called Archaebacteria. Now, as you know, archaea means old. Okay, so these are the most primitive type of bacteria, and they are able to live under very extreme conditions. Then the other group is called Eubacteria. As I mentioned earlier, Eu means real. So these are considered the real bacteria. And the other Archaebacteria are considered the older version or older generation, or the what we call a primitive type bacteria. Okay, so you just remember now here, just want to try to help you to remember. Prokaryote, you have only two types, and they are all bacteria. But only thing, you have a older version of bacteria, which is called the Archaebacteria. Archaebacteria, archaeology, right? Archaebacteria. The other one is a Eubacteria. So these two are prokaryote. They do not have membranes around their organelles. Okay, they have circular DNA. So the other four kingdoms, they belong to the Eukaryotes. Okay, they are the organelles are membrane bound. They have linear DNA. So what are the four here? These four we find it more common that we know of. These are the examples that we know of. Protista. Protista are those single unicellular organisms that live usually in wet environment, in the water, in the lakes, like amoeba, all right, and paramecium. These are examples of that. Okay, Protista. And also the type of organism that causes malaria, they're called Plasmodium. So they are under Protista. They are single cell. They are unicellular. Then fungi. Fungi can be unicellular. They can be multicellular. Okay, so examples would be the yeast that you use to make bread, and also all the types of mushrooms. The mushrooms and the molds, the bread mold and the mold that you find on your old leather jacket in the cupboard, right? Being kept in too clammy conditions, too wet, they will start to become moldy. So all the molds, all the mushrooms, all these yeast and so on, they can be, they are unicellular, but they can live in colonies, right? They are fungi. And they don't have chloroplasts. They are never green. Mushrooms, as you know, they are never green. They're either gray or white or black, right? And they do not photosynthesize. They actually they're saprophytic, so they actually absorb nutrients from external environment, from the outside of their host. Okay, they don't make their food. Now, Plantae, this is quite common. These are all plants. Generally, these are all the plants that you see, the green plants, the plants that have chlorophyll, that you see all the mango trees, papaya trees, durian trees, all the grass and the flowers and so on. These are Plantae. And Animalia is self-explanatory. So Animalia are all the animals. These animals can move around. Okay, so basically I've given you a general outline of the 6 kingdoms so that you can see an overall picture. Okay, which type are prokaryotes and which types are eukaryotes. All right, okay, so you get a clear picture. You may want to jot this down, or you can play back later, all right, this video. Okay, now let's just compare the prokaryote. So this is picked up from the internet. So again, the most important thing is you say that they are either membrane bound, it has membrane bound nucleus, then you'll be called as eukaryotes. Now, what's the difference between eukaryotes and eukaryotic cells? Actually, they are the same thing. When you want to talk about the cell itself, you say it's eukaryotes, full stop, can. It's referring to the cell. If you want to say the word eukaryotic, the word is an adjective. You cannot say eukaryotic, full stop. You have to say eukaryotic, noun. A noun is the word cell. So eukaryotic cell. Okay, so if you say eukaryotes, it's okay, fine. It's the noun already. The word is a noun. But eukaryotic cell, because it's an adjective, you cannot just stop at adjective. Adjective must be followed by a noun. So eukaryotic cells. Okay, so then what about prokaryotes, or we call prokaryotic cells? They are not membrane bound. Okay, then the eukaryote is linear DNA, while the prokaryote is circular DNA. Okay, so if you will see in terms of picture diagram, right, you will see very clearly for eukaryote, you have a nucleus which is bound by the membrane. So you can draw an outline there before you start dotting or stapling your nucleus there. But for the bacteria, which is prokaryote, when you draw your nuclear material, you don't draw a line around it. You just start to, just make it more dense to show the nuclear material there. Okay, so remember, do not draw a membrane around your nucleus for prokaryotes. Okay, so that is the main difference there, all right? And of course, the DNA shape lah. Okay, so here, let me go on a little bit more. So in all the cells, whether they are prokaryotic or eukaryotic, they should have all these, okay, all cells. They have DNA, they have plasma membrane, cytoplasm, they have ribosome. So this is additional material for you. And what are the difference? Again, so I put it in diagrammatic form here. Nucleus, okay, we'll say nucleus is not the real nucleus. That's why they call it absent. Okay, for eukaryote is the real nucleus, so we call it present. Okay, you can ignore this one. All right, you can ignore this one. You just mentioned prokaryote do not have membrane-bound organelles, but eukaryotes have membrane-bound organelles. Okay, so they are usually unicellular. Okay, mostly eukaryotes, most of them are multicellular, like all the others, the animals, the plants, and all that. Most are multicellular. Cell size, prokaryotes are generally smaller, but eukaryotes are generally bigger. Complexity, prokaryote, of course, because they are primitive. So primitive means they are simpler, definitely. Okay, eukaryotes, they are more complex. DNA form, circular, linear. And examples would be these two type of bacteria, the Eubacteria, which is the real bacteria, and the Archaea Bacteria. And for eukaryotes are the other four groups, other four kingdoms. So there you have 6 kingdoms. Okay, you need to know what is prokaryote, what is eukaryote first. Okay, now we go into the this one. What are these 6 types of kingdoms? We go into Archaebacteria, Eubacteria, that's for prokaryote. Then we proceed with the eukaryote. Okay, any questions here so far? Let me just check on comments. Okay, so far nothing. All right, okay. So here are this diagram now, okay, which I've got from the internet. This is Archaebacteria. It's about the same diagram that we have. Here are characteristics of Archaebacteria. Okay, let's start with Archaebacteria now. Okay, here. So Archaebacteria and Eubacteria, as you know, they are prokaryotes. Okay, and the other 4, Protista, Fungi, Planta and Animalia, they are eukaryotes. So prokaryote means it's a primitive nucleus. Before, pro means before, it does not have a true nucleus, so you don't draw a membrane around it. There's no nuclear membrane. Okay, and then the eukaryote has a true nucleus. There's a nuclear membrane. Okay, usually, actually, last time in your KBSM syllabus, your seniors, we don't have 6 kingdoms here. Last time, according to the KBSM, there is only 5 kingdoms. They combine these 2. Last time in your KBSM, just for you to know, okay, they have separated this one. Last time when your seniors, the KBSM syllabus, these two are put under the same group, which is called Monera. Monera, M-O-N-E-R-A. Okay, they are put in. But now a scientist has discovered that there's a difference in the shape of the DNA. So one is circular, the difference in the composition. Okay, so they have separated the Archaebacteria and the Eubacteria. So here you will see this note here to show. This is also in your textbook, to show that it's a new division. So Robert Whittaker, 1969, we introduced the 5 kingdoms, which is Monera, Protista, Fungi, Plantae, Animalia. So the KBSM syllabus, we use 5. But now, developments in the study of biomolecules found the differences in the RNA between the bacteria and the Archaebacteria. So that's why they proposed this. Carl Woese proposed that this Monera be split into 2. One is Eubacteria and Archaebacteria. Okay, just a background for you. And afterwards, you will see this term as we were discussing the Archaebacteria and the Eubacteria and the other eukaryotes. We are going to see these terms, okay? Autotroph and Heterotroph. So I want to explain first what is autotroph, what is heterotroph. Autotroph refers to, this is referring to the organism or a living thing that are able to make its own food. That's very simple to put it in that way, okay? All the organisms who are able to make their own food, they are called autotrophs. So very simple, like your plants, your green plants. Okay, and also some bacteria are able to do that. They either use the light energy. So for plants, green plants, they will use the light energy, the light from the sun. They carry out photosynthesis. Or another group, they also use energy, but not light, but they use chemical energy to produce energy. Okay, to produce a food. So this is autotroph. Autotrophs are organisms which make their own food. So they are able to make their food or produce more complex substance from the inorganic substance. Okay, and for heterotroph, Hetero means many, many types, okay? These organisms that cannot synthesize its own food, just like us, we belong to heterotroph. So as same as like any other animals, cows, okay, dogs and all that, they cannot make their own food, but they have to obtain the food from eating other organisms, okay, either other external source. Not necessarily eat, they can also absorb. They can also absorb from another organism like parasites, okay, or saprophytes. They can also absorb. They don't have to necessarily eat into the body. So these are called heterotrophs. So basically, either you can or you cannot produce your own food. They are classified as the autotroph, you can. Heterotroph cannot produce food on its own. Okay, so these are the two terms you need to understand because we're going to see it afterwards. Okay, so now we go straight to the 6 types of kingdoms. Okay, for today's lesson, I'll just stop at 6 types of kingdom because this one has a lot of terms already. Okay, so it needs time for you to digest. So I'm not going to go through further. The other things I'll go through on the next lesson, how to name it, how to classify that. It'll be another lesson because it's quite a bit for you to digest here. There are so many new terms for you. Okay, right. Let's go to Archaebacteria. So here, the notes here are quite limited. Okay, so I've used these notes from here. Okay, so here, these notes are a bit less. So I've got added notes here in this on this page here. So let's look at it. Now, again, Archaebacteria is prokaryote, so you need to remember the characteristics. That's why just now the diagram there helps you. The what they call the chart here to see which domain or which kingdom comes under which, whether they're prokaryote or eukaryote. Okay, so prokaryote, okay, they are prokaryote, they are unicellular, so bacteria are always unicellular. They are primitive because of the archaea means they're old type, okay. They can be autotrophic or they can be heterotrophic, okay. There's no true nucleus because there's no nuclear membrane, okay. They have cytoplasm, okay, which contains all the compounds necessary for their metabolism, and they have a circular DNA, okay, which they call plasmid. Okay, no membrane-bound organelles. So the whatever organelles they have there, they are not they're not bounded. They don't have a membrane that covers it. Okay, now this is one of the facts you need to remember. It has no peptidoglycan. So what is peptidoglycan? Peptidoglycan is a substance which is made up of actually a combination. Inside there you have sugar, from the word glycans means sugar because glycose, okay? Peptido is protein. So it's a substance that is basically contains sugar and also protein. So it's called a peptidoglycan. It doesn't have, the cell wall does not have peptidoglycan. Peptidoglycan, another name is called murin. Okay, so these two are same. Murin is another name for peptidoglycan. This cell wall, so remember this cell wall is not cellulose. Cellulose is only for the plants. Okay, so even though it doesn't mean always you see the word cell wall, it must be cellulose or cellulose. Yeah, no, it's not cellulose. For bacteria, it's not cellulose. For bacteria, it could be peptidoglycan, but Archaebacteria, no peptidoglycan. Okay, the cell wall is to give support, all right, give shape. So as usual, it is rigid, so it gives support. It prevents the cell from lysis. Lysis means bursting during hypertonic conditions. That means water will rush in. Okay, they usually carry out asexual reproduction by binary fission. That means from one cell it's split into 2, from 2 to 4, 4 to 8, and so on. So it's a very fast way of reproduction. Okay, usually every 20 minutes you get a new generation. So that's why food can spoil very easily once you put it into room temperature. As long as you have bacteria there, every 20 minutes, it will actually multiply. The number of colonies, a number of individuals of bacteria there will actually multiply every 20 minutes. So it's always important to keep it refrigerated, okay? Food that that you do not eat. Right. So able to live in extreme environments. Now, these Archaebacteria are very special features they have. They are 3 groups here under the Archaebacteria. They are able to survive very extreme extreme conditions that other bacteria or even other organisms cannot survive. Example, what are the 3 extreme conditions? Number 1 is very salty environment, high salinity. Okay, this group of Archaebacteria are called halophile. Okay, halo, as you know, halogen, it actually means salt. Okay, so halophytes, you've learned the previous lesson. Halophytes, these are plants, phyte, P-H-Y-T-E. These are plants which live in very salty conditions like the mangrove swamp. So halophile, organisms that live in very salty environments such as the Dead Sea. So you have these bacteria inside the water there, which have high concentration of salt. So they're able to survive. They do not undergo plasmolysis. Okay, so they won't die. Another type is called Methanogen. Okay, so these are the group of bacteria, Archaebacteria, which live in conditions which are very low oxygen. Okay, they're very low oxygen. So these kind of bacteria are called obligate anaerobic bacteria. What's obligate? Obligate means it's like compulsory. They have to live in conditions which are very low oxygen, because high oxygen content actually may even kill them. Okay, so they have a very peculiar, maybe they are have a different metabolic pathway lah. So they do not operate in the same way as all organisms do. So they have to actually live in places or conditions which are very low oxygen. So they are obligate anaerobic. You learned earlier last year. Anaerobic means conditions which are lack of oxygen. So they are found in swamps. Okay, swamps with low oxygen content, and digestive tract of ruminants. Ruminants are those animals which eat grass, like cow and so on. So in the stomach of these cows, they have this kind of bacteria. So they have low oxygen levels there. And also in our gut, all right? And as a side effect of their metabolic process, they will produce methane. Okay, so in our gut, in our intestines and and our intestines and so on, we will have actually some bacteria living there. And as one of the metabolic reactions there or processes, they will produce methane. So in our fart, okay, our fart, we call it kentut. When we kentut, we pass gas, right? Okay, excuse the term farting. It's actually passing gas. The gas actually, one of the gases that are inside our that is inside there is actually methane. And as you know, methane is actually flammable. So our fart is actually flammable. I don't know whether you know that or not. Okay, if you were able to collect your passing your gas, you collect it in a substantial amount, you were to set a fire to it will actually explode. Okay, so this is something peculiar, something funny, but actually it's the truth lah. All right, so it's something for you to know. I like to tell you all these things when I'm teaching, so you get a lot of info. Everything is interrelated. You're talking about methane, you also can talk about our gas in our body. Okay, so these methanogens are bacteria which are able to survive in low oxygen conditions. Okay, thermal acidophile. So thermal acetophile. So thermal, as you know, is heat. They are able to withstand high temperatures. Now, these are the type of organisms, for example, we see in hot springs. Okay, the water is above 60 degrees Celsius. Sometimes when we go and dip into the hot spring, we even find our skin is all red. And then some of us are not able to, we don't like this kind of too hot conditions. We find it very hot, but normal organisms will even die because, as you know, right, enzymes will denature. After like 50 degrees Celsius, most of the enzymes will denature. So the normal organisms cannot live there. But these are special breed of bacteria we call Archaebacteria. They can survive. In fact, this is their environment. They live in very hot conditions. Okay, and they also have high acid tolerance. They can also live in conditions which are high acidic conditions. Okay, so in the hot springs like a geyser, one of those national parks where we have all these hot water geysers and all that, they actually exist bacteria there. Okay, they're called thermal acidophile. Now the meaning foul, all right, the word foul means they like the condition. They are there's affinity to it. So like for example, you heard these names, all these words, pedophiles. Pedophile are this very psycho people who really, they prey on little kids. They like little kids and they want, they will adopt kids and then they will do very bad things to the kids. They are called pedophile, isn't it? Foul actually means they like. Okay, so this is the meaning of foul. Foul, they like this. Halophile means they like salty conditions. Methanogens, this one doesn't have the foul. The thermal acidophile, they like all this hot and also acidic environments. Okay, so they will use flagellum to move. Flagellum is actually this thing. Usually bacteria will have some kind of a strand of this thing jutting out there. Either it could be single, it could be double. So they will help them to move around. And the short ones are called cilia. Okay, the long one is called flagella. The short one is called cilia. Cilia, all these things here. You see all this? So all around the surface, you have cilia. Usually all this is to help in locomotion, to help them to move. Okay, so we go on to the next one. Eubacteria. Okay, now Eubacteria, this is is also prokaryote, but this is a true bacteria because, okay, there's also no nuclear material. All right, but the difference here is, they have peptidoglycan. So the difference between Eubacteria and the Archaebacteria is they have peptidoglycan. Okay, peptidoglycan is also known as murine. So it's a combination, it's a substance made of combination of sugar and also amino acid. Okay, so this basically there's a difference between your Eubacteria and your Archaebacteria. Both are prokaryote. Okay, both do not have they are not there's no membrane to bind their organelles. But the difference here is one has peptidoglycan, the other one does not peptidoglycan. Okay, this flagellum for movement, very similar to that. They can be autotroph or heterotroph. They carry out asexual and also asexual reproduction by binary fission and also budding. So they can also carry out budding. Okay, and they can form spores. They can stand extreme conditions. So when there's lack of food, they will just become dormant. They will just roll up into a ball. Actually, not roll up in a ball. They just have a protective shell over them, and they will just stay in that condition until the condition becomes favorable, and then the bacteria will live again. Okay, so examples of Eubacteria are those bacteria that we commonly see. All right, these are the very common examples here. Staphylococcus, okay, Bacillus is a rod shape. So for this Eubacteria, they are classified according to their shape. Okay, so you have coccus. Coccus means they are round shape, like just sphere. Bacillus is rod shape. That means they are long in shape. Spirillum, they are like ribbon. They are spiral. And Vibrio, they have the shape of like a comma there. So there are 4 groups under Eubacteria. All right, so they are classified according to the shape. But for Archaebacteria, they are classified according to the habitat. So you have 3 groups there: halophile, methanogen, and thermoacetophile. This is based on their habitat. Okay, so I want to show you. I think I've got some information on the differences between the 2 types of bacteria here. Okay, Archaebacteria and Eubacteria. So Archaebacteria, no peptidoglycan, no nucleus or organelles. They are unicellular. Some are heterotroph, some are autotroph. So the difference here, okay, you don't have to be very specific about this. You just look at the part which I just highlight. So they are both single cell. Okay, but the difference here is archaea, they live in extreme conditions. For Eubacteria, they live in normal conditions. They're called the true bacteria. Both, last time they call it Monera kingdom. Now they split into Archaebacteria and Eubacteria. They are different kingdom considered. Last time they called Monera. Okay, where do they stay? They are unusual environment. For the archaea, you have very ocean deep, at the very deep layers of the sea here. Okay, volcano sites where it's very hot, salt brine, that means it's very salty condition, and also very low oxygen conditions. Okay, Eubacteria are found everywhere, like in the soil. This is the common bacteria that we see around us lah. Okay, so in the soil, in the water, in the air, and so on. So reproduction, they happen by binary fission. Okay, sometimes budding process, but usually binary fission for Archaebacteria. So they also can produce sexually for Eubacteria, sexually and also asexually. So they have 2 types. They can actually produce sexually and asexually. So the difference here in the cell wall, one is peptidoglycan. This is no peptidoglycan. This is called pseudo. Pseudo means false. Okay, pseudo means it's not the real type. So it's pseudopeptidoglycan. For the Eubacteria, it's peptidoglycan. Okay, one more difference is Archaebacteria are usually not harmful. They are not known to cause diseases. Okay, but Eubacteria, some of them are pathogens. They can cause diseases in humans, like for example, Escherichia coli. Okay, so they are able to cause diseases. So they are pathogenic. Okay, let's look at the next. So we've already covered 2. We already covered 2 groups here, or 2 kingdoms here. Archaebacteria and Eubacteria. Okay, so you can refer to your textbook later. These are some examples lah. Okay, examples. sulfolobus. This is a sulfur oxidizing bacteria. These are the autotroph, okay, autotrophs, and they're able to produce their own food, but not using light energy. They use the different type chemical energy. Okay, Halobacterium. This is the ones that are able to stay in very salty condition. Halobacterium. Okay, so the other one, Streptococcus pneumonia. This one causes pneumonia. Okay, infection of the lungs. This is the type of bacteria which causes infection of lung, pneumonia. And Vibrio cholerae. This is the bacteria which causes diarrhea. Okay, cholera, diarrhea. Cholera, actually cholera. Okay, we call it penyakit taun in BM. So they are known to cause diseases. Okay, next one. So let's look at Protista. So we're coming to more and more common. These are all eukaryotes already. The first two prokaryotes are already finished. Now eukaryote. Now we're talking about the eukaryotes. Okay, Protista. They can be multicellular. They can also be unicellular. They can be autotroph. That means they can produce their own food. Okay, or they are heterotrophs, or can also be both. And most of them, they form colonies. That means they live in groups. For example, yeast. Okay, yeast, they will multiply through budding, and they will form colonies. Okay, so they will have the true nucleus. Of course, they have the membrane. So whenever you draw these diagrams, you must draw a membrane around the nucleus. Okay, they also have membrane-bound organelles. Everything is membrane-bound, nucleus and organelles. Okay, so possess cytoskeleton, so they have something to make it rigid. Okay, there's a network of proteins. Okay, not so important this point. Some of them are able to move. They're able to swim in the water. They have flagella or pseudopodia. Like for example, the amoeba. Amoeba, they will glide along by spreading the cytoplasm, right? And then they project the pseudopodium, and that's how they eat. But they engulf the food and bring it into the cell itself. They carry out asexual, or they can carry asexual, or they also can carry sexual. Okay, paramecium are known to carry out sexual reproduction. They can exchange genetic material between two parameciums. They are able to exchange genetic material. Okay, then Protista can be divided into two groups. Okay, you have protozoa. Protozoa are those examples like amoeba, paramecium, euglena. Algae would be the Spirogyra, Chlamydomonas, volvox, that. Slime mold. Okay, slime mold is another one. Okay, so now next group, eukaryote, also eukaryote. We're all talking about eukaryote now. Fungi. Now, fungi is all those groups, all those organisms which are does not have chlorophyll, but they look like plants. For example, like mushrooms. Okay, mushrooms, they are fungi. Okay, they are heterotrophic because they are not able to produce food. Okay, so always remember, mushrooms, you never see them as green. Okay, you never see mold as green. So what they do is they have to get it from somewhere else. Usually they will absorb it from whatever they're staying. So for example, the bread mold. So you have the mold staying on top, right? It's actually drawing nutrient from the bread. So all these what do you call these strands? All these strands that come out. This is a bread mold. So imagine this is put under a microscope. You have the white color lines. When you see a bread is molding, you'll see all these lines there, right? The white color. And then all these are called hypha. Okay, so when you have a network of it, you call it mycelium. So all you have those strands here, the filaments, thread-like structure, they're called hypha. And when you have a lot of this, you form a network we call mycelium. So when they become mature, they're able to produce spores. And when the spores are matured, they become black color. So that's why you see the moldy, sometimes green color as well. You see all the mold on the bread, right? And then they will start to erupt, or they burst. This sporangium will burst, and the spores will be spread out. And that's how they disperse their spores, and they grow more and more. Okay, so they are heterotrophs. To be specific, they either suck or absorb the food from outside. They are called saprophytes. Now, all this specifically we'll see in another lesson. There will be a separate lesson to see all these terms. Okay, so I do not want to give you too many things today. I just want you to know the 6 kingdoms only. Okay, so another group will be parasites. They will live on. What's the difference, saprophyte and parasite? Saprophyte is this organism, this mold will live on the organism, or the host is already dead. So the saprophyte, that thing they're living on is already dead. It's not a living thing. Okay, example, what do you call rotting tree, the tree that is already rotting, already dead. Okay, or you can eat on the bread. Bread is not a living thing. All right, so it is absorbing food from the bread. That is the bread mold or mucor. Another name of this is mucor, right? But separate for parasites, if you're talking about a mold that is living on a parasite, for example, I may have, let's say some people have all this itchiness right on the skin here, right? It could be fungi, all right, staying here, and you are alive, isn't it? So this is a parasite. Okay, sometimes a parasite is an organism which actually they are living on the host, and the host is alive. So it's called a parasite. Okay, so it digests the food externally and then they will absorb it into their body. So they live in moist and hot place, not to say too hot lah. Okay, if it's too hot, extreme condition, they will not be able to survive. But most importantly, they need to be have moist condition. So therefore, if you do not want your shoes, your leather shoes or your leather jacket to become moldy, you make sure you have a dehumidifier in your closet. Okay, or you put something to absorb the moisture, then you will not be growing fungus. Okay, so they carry out asexual reproduction by usually spore formation. So sexual reproduction is very rare, okay, for fungus. Okay, now Plantae, this is more common to you all. These are all the green plants that you see, even moss, okay, anything that is green, because they all have chlorophyll. They are eukaryotes, okay, multicellular. They are immobile, they cannot move, they are sessile. Okay, they are stuck there. Okay, another word is immobile, is called sessile. They are not able to move around. They are autotrophic, that means they can produce their own food. They have chlorophyll. Okay, and this type of organisms which are able to produce food using the light from the sun is called photoautotroph. Photo means light. Autotroph means producing their own food. Using light to produce their own food is called photoautotroph. Okay, now for this Plantae, the cell wall is cellulose. Then you see the cell wall is not necessarily cellulose. For the bacteria, it's never cellulose. It's either you have peptidoglycan or don't have peptidoglycan. Their cell wall is peptidoglycan for bacteria. But for plants, okay, those green plants, especially the cell wall is cellulose. For the fungus, the cell wall is chitin. Look at different material. So that's how you classify them because of the characteristics. If they have chitin, it cannot be a Plantae. Okay, so if that you let's say you discover something new, you discover a new species, and you do some investigation and you find out certain features. Okay, you do not know what it is. So first of all, you got to see the features. Okay, let's say you discover, okay, I tested, okay, this looks like, this is cellulose. So with cellulose, right, it cannot be a bacteria. It cannot come under fungus. It must come under Plantae. Okay, so that's why these are the basic characteristics that you need to remember in order for them to be classified as such, as an organism in that group. Okay, so it has cellulose, okay, and they carry out, it could be asexual, it could also be sexual. So like for example, ferns, they have spores. Okay, for flowering plant, they have the male part, which is the pollen grain, the male gamete, and you have the embryo or embryo sac with the ovule in the female. So you have both. And then they are also classified into seedless plants and also with seed plants. Okay, so flowering, non-flowering. So this is very wide. Plantae is a very wide group there. Okay, lastly, we come to Animalia. Right, Animalia is of course eukaryotic. You have many types of species, especially ants, a lot of types of ants, and also insects. Insects is one of the groups that you have many species under it. Okay, even more than other mammals. Most animals can move around, so they are not sessile. They are able to move, and they are heterotrophic. Definitely, animals are heterotrophic. They cannot make their own food. They rely on the energy that is already stored in the form of starch, right, in the food, in the plants. So their cells do not have cell wall. Okay, definitely one of the characteristics, and there's no chlorophyll. So if you see that this thing does not have chlorophyll, you cannot put it under Plantae. Okay, so you got to look at the features. And they will mostly carry out sexual reproduction. So these are under Animalia, you have 5 classes where you have your mammals, reptiles, amphibians, bird, and fish. Okay, and another group here you call the invertebrates, which don't have backbone. So under Animalia, first of all, you have chordata, or chordata means we call it with backbones. Then you have another group without backbones. Okay, now here, interestingly enough, look at this sponge. Sponge, I think a lot of people do not know, a lot of students do not know, sponge is actually an animal. It is not a plant, okay? It is not something that you grow. It is an animal. Actually, this is the sponge that is naturally, this is a real sponge that you get from the sea. I'm not talking about the synthetic sponge that you buy from the shop. Okay, that one is urethane. That means man-made. That one you buy from the shop, you use it to wash your dishes, right? That's a normal sponge. That is man-made. I'm talking about the real sponge. The origin of the sponge is actually from an animal, which are still in the sea there. Okay, so these are the real sponges, and they're very, very porous. There are a lot of holes there. So actually, they are an animal. Okay, it's not an organ, it's not a plant. All right, okay, so this is something new, maybe for most of you lah. Okay, so I have covered here 6 kingdoms. Okay, so here I will not go further because it's quite enough for you to digest. There are so many words, so please go through your textbook. Okay, go through whatever workbook you have or whatever reference book you have. Make sure you understand every term, because the next lesson will draw upon this information also, where we go on to further classification after your kingdom. One kingdom is a big group. You have to separate out into smaller groups. So we have a different name under that. Okay, so you have different levels. Kingdom is one of the highest levels. Then you under kingdom, you have many groups also. Okay, so let me see. So today no questions, I think. Something very new, I think. Probably nothing, don't know what to ask also, right? Okay, but don't worry. In our lesson next week, I will go into our Google Meet and we can have discussion here. I can repeat certain things that you don't understand, or you can ask questions. Okay, so I would like, for those students in my class, I will give you this as an exercise in the Google Classroom. Okay, I want you to fill it up. You need to digest the information and you fill it up, all right, and you send it to me. Okay, I will make that worksheet for you soon, okay, within this 1 or 2 days. Okay, so this I will leave it for the next lesson, the taxonomy hierarchy. How do we have these different levels? Okay, so the highest one is the domain. So we have found that there are actually 6 kingdoms there, right? Then after that, you can further divide. Okay, right. So let me check. No questions here. So it's almost time, right? I hope you have understood the lesson. So I had to do a lesson today because there are a lot to cover actually. We may not have enough time. So I want to just carry out this lesson first, and you have something to work on next week when we start our lesson. We have our Google Meet, then we can have discussions there. Okay, right. So I will stop sharing now here. Okay, so I hope to see you next week, right? I might have a lesson. Usually this could be quite a fixture, like maybe every Saturday, 4 o'clock. So I will send the link to you. And for those who are out there, if you have subscribed, then you will also get the notification. All right, okay, thank you. You mean you're welcome. All right, thank you, girls. So I will see you next week during class. Okay, right. All right, bye-bye. Bye-bye, girls. Bye-bye, students out there, whoever you are. Okay, bye.